All right, let's get started. As I mentioned before, so as the poll's going, we'll probably be running through a very brief introduction about BioRender, about really what we're trying to do in this space, in the scientific communication space. And then I'll jump into a very brief kind of introductory demo of basic and maybe intermediate functionalities and content inside BioRender application. And then I'm going to send the file over to Shiz, who will take it from there. Okay, great. Thanks everyone for inputting votes into the poll. I guess they're not really votes, but into I guess a bit about yourself into the poll. It looks like we have a really cool spread of attendees from inside in molecular biology, biochem, biotechnology. Oh, interesting. Immunology, and actually quite a few attendees are in the, I guess what we call the other field. So if you'd like to type that out in the Zoom chat, that would be great. Thank you. And it looks like we have a vast majority of our attendees today have not used BioRender, but have either signed up or not signed up yet, and a couple have used it once or twice. And we do have potentially one attendee who is a BioRender veteran. Hopefully we'll also have a bit of, maybe a bit more subjective design, I guess, wisdom or tips to share throughout this webinar that will be beneficial for you as well. Thanks everyone. I'm going to end the poll now. Thanks for inputting your feedback, or I guess not feedback, but answers. Okay. So a bit about ourselves. As Shiz kind of popped her microphone in a little bit earlier, Shiz, if you'd like to jump in again and provide a bit of an intro about yourself.
Yeah, sure. Thanks, Jerry. So my name is Shiz. If we haven't met already, I'm one of the co-founders of BioRender, and my background, very similar to Jerry, is in medical and biological illustration.
Thanks, Shiz. Yeah, and as for myself, at BioRender, I'm primarily leading the template and content design team, but also I'm a UX designer in BioRender, and kind of inputting feedback and providing guidance in terms of various application functionalities. And in terms of my background, I actually come from more of a research background. I did a master's in biochemistry before switching over to medical illustration, both at University of Toronto. Okay. So I guess as Shiz mentioned, her professional career is in the science communication and illustration field a bit before my time. And back then it allowed her to lead the science illustration team at National Geographic for about a decade. A lot of us more junior science illustrators are big fans of hers from National Geographics, of all these beautiful illustrations you've made back in the day. And as we knew also that you worked extensively with scientists and with popular academic journals such as Science and Nature. There was a bit of an identification then that really a lot of scientists and labs don't have the budget, the resources, or the connections to collaborate with professional medical or scientific illustrators like ourselves to visually translate their science. And that's a big problem. We're a very niche field, I guess. And because of that, a lot of scientists end up using PowerPoint or more convoluted programs like Adobe Suite to spend hours, pouring hours into making illustrations, trying to articulate their work. Here are just a couple of examples of one such instance of different scientists or labs trying to visualize a foundational immunology concept of some cells presenting antigens to some other immune cells. And unfortunately, you see dozens, sometimes hundreds of different ways of showing essentially the same pathways or reactions or methodologies. Some of them are fairly successful in communicating their content, but many don't quite make the cut. So back then, Shiz and some of her colleagues asked the key question, what would happen if we as science illustrators, provided all the little graphical elements involved in science figures as a standardized set inside a really easy to use tool? Would the scientific community find ways of putting them together into their own compelling, clear illustrations? And that's really how, I guess, BioRender got started. So here at BioRender, we can't really claim to fix this problem of effective, standardized, easy science communication holistically, but we do hope to help at least in two regards. One is making the whole process easier and faster, and two is to make the end result much easier to consume and clearer. Here's just a speed through GIF of a few of the capabilities within BioRender. A little bit sped up, but of course, we'll pick this up as we continue through the actual demo portion. Here are a top list of really just some use cases of BioRender that our user base has been able to do. I would say that the kind of major use cases are things like figures for publication, for various presentations, bigger talks all the way to smaller lab meetings, as well as figures for posters and such. But the list definitely grows over time as our users find more ingenious ways of visualizing and communicating their science. Also, I mentioned that we have templates in our library in BioRender, and that's one initiative I lead and spend quite a bit of my time on. Here are just a couple of examples. Our templates cover pathways, clinical figures, experimental methods and protocols, graphs, timelines, etc. And we're really adding more every single day. And it's also a great way for us, I guess, to showcase the breadth of topics that we hope will strive to cover in the bigger life science kind of domain ecosystem. We also try to tackle both more niche fields, more specialized fields, as well as more general kind of more heavyweight fields like nowadays, like immuno-oncology and things like that. So with the template library, it's easy for you to search for and find the templates you need. Some of them are almost complete figures. So if you find something that's almost exactly what you need, then you will be able to just jump in, change a few things, maybe remove a few unneeded things, and finish off with a really clear, concise figure of the knowledge you're trying to communicate. Otherwise, some other use cases here might be to take small pieces of certain templates and build your own. Just to wrap up my brief introduction here, our goal at BioRender is to give every scientist both the tools and the practical knowledge to visually communicate their research. Now, with that in mind, our aim today is to showcase some basic as well as some more advanced capabilities of our tool. And throughout that process, hopefully to provide some general practical tips on designing your own effective figure. Okay, so on that note, I'm gonna hop over a window and jump into BioRender and have a go. Okay, hope everyone can see my new window here. Here's just a demo account where I'm kind of landed on the gallery page here. This is where your central kind of file hub is. And as you make more and more new illustrations in BioRender, the bottom part here is going to populate. And if it gets a little bit too much, you can set up new folders and drag them in and organize your files as you need in BioRender. But along the top here, you can see that you can start either from a blank illustration or from one of our feature templates here. The feature templates right now are a little bit arbitrary. We kind of choose them based on some new templates we would like to show off, or based on some important subject that's really relevant in the research community right now. For instance, here we have the coronavirus replication cycle. But for just to start off our demo today, I'm going to start with a blank illustration in the top left here. Click that, and here I'm just going to assign a figure name. and choose from one of the size presets. This is, you can always edit this inside the editor at any point. So I'm just going to go with a default, which is a slide size. Create illustration. Okay, so here we've landed on the what we call the canvas view or the editor view of the BioRender application. And there's quite a bit of user interface here. I'll just kind of breeze through it first before we jump into any design. Along the left-hand side here, and quite prominently, is kind of our content insert pane. And here you can insert from our huge icon library of, I think, about 40,000 professionally curated and designed scientific icons now. You can navigate through any of these categories and browse as you need, or as most users would like to do. If they know what specific thing they need, they can, basically input into the field on top and see the results. I'm going to clear the search field. And along that line, on the left-hand side here, you can also toggle between different tabs to access other rich content that you can add to the canvas. But I'll go through those in a bit. In the middle top here is the toolbar, and this is a contextual toolbar. And what that means is that you have some basic things like inserting shapes and lines, some canvas options. But as you have content inside the canvas, let's say if I put an Amazon parrot here and I select it, the toolbars is going to responsively reveal customization options based on what I select. On the right-hand side here, you have a bit of navigation tools. So, zoom, pretty standard stuff. And ruler, so you can show ruler, hide ruler. For those who like to add grid lines, the revealed rulers allow you to add these guides that allow you to kind of do more precise spacing and gaps like that. I'm going to clear the guidelines. I'm going to right-click on the ruler and clear all guidelines. And at the bottom-hand side here, you have a few other things. Keyboard shortcuts for those who like keyboard shortcuts and like to speed things up that way. And FAQ, as well as our amazing customer support team that you can access and talk to via this button here. And on the top right, you have a few other functionalities like preview, export, and share with. Okay, so I'm going to unfortunately delete this bird. And I think just for our demo, kind of first portion of the demo today, I'm going to put together a really simple cell-cell interaction diagram and along the way showcase some functionalities. So first things first, let's put in two kind of big zoomed in cells. So I'm just going to search phospholipid bilayer. And pick something that works. Let's see. Yeah, let's try this one. So membrane phospholipid bilayer medium filled. I'm going to drag it in. And now that it's in, I can drag the side handles to just make it a little bit bigger. And then for the top cell, I want to make it look like it's kind of coming in. So maybe I'll use one of these ones, arc membrane. And now that that's in, I'm going to drag the rotate handle to rotate it. It's going to snap in to the 90-degree rotation like that. And I'll just kind of frame it more along the top here and make it a bit bigger so it goes off the canvas. And that's important so that if I click preview, you can see that there's no white gap around the edges. I'm going to move the bottom membrane up a bit more, make it a bit bigger. Perfect. Like that. Okay, so now you have a nice kind of background interface to show cell-cell interaction. I'm going to style them a little bit. So the top one, if I click it in a toolbar, icon color is revealed, and I can just kind of choose from a number of different icon colors. Let's go with red for now. And with the bottom cell, maybe I'm going to go with more of a blue color. But this is actually we're in the process of making our icons more customizable. So the top is a simple icon, it has a number of presets that you can choose from. The bottom is actually a more customizable icon because it says here, double click to edit group. It's a grouped icon. And what that means is I can double click, and now I'm inside the group, and I'm able to edit different parts of this icon. So I'm gonna click the phospholipid part on the top, click icon color, and let's go with more of a, what's good? Yeah, this nice bright blue here. And as for the bottom part, this is a simple shape. So you can go to color presets. Let's go with a nice, clean, vibrant blue. I'm gonna click away, exit the group editing mode, and there we go. One last thing I'm gonna do here before I jump into adding some major players is just to multi-select them. So I'm gonna hold down Shift on my keyboard and click again to select them both. And in the toolbar, I'm just going to lock object. And as you see here with the little tutorial that popped up, it allows you to avoid editing that object when you try to illustrate. So this is great for backgrounds. Got it, thank you. So if I lock object now, boom, you can no longer accidentally drag them. This is a good place, I think, to pivot to our next point, which is we're going to insert some proteins where they interact. So let's say the top is an antigen presenting cell and the bottom is a T cell that's receiving the antigen. So let's do TCR for T cell receptor. And that's pretty good. So an MH2 antigen presenting antigen to a T cell receptor. So I'm going to drag that out. It's also a grouped icon, as you see in the icon library with a purple border, which means it's a hint that you can edit individual pieces separately. So I'm just going to scale that down a little bit so it kind of fits. Perfect. There we go. Oh, and incidentally, I guess the colors match because I made the top red and the bottom blue. That was not planned. But if you still need to edit the colors, of course, you can double click in, or you can, if you don't like double clicking, you can right click and enter group. And let's make the top a bit more orangey. There we go. Perfect. And a couple of tips here. So let's say if you want to make this scene a bit more dynamic, you can duplicate these features so that it feels like a more lively environment. And maybe if you need to show some other interactions here as well. So I can click that, hold down Alt key or Options key if you're on a Mac, and then just drag, and that's going to repeatedly duplicate as long as you hold down the Alt or the Options key. So I'll just do that for a couple of these. Maybe I'll delete this one, like so. Actually, I don't need these ones. There we go. Okay, so let's say now you kind of have the scene set up and you'd like to add a bit of a downstream effect in this interaction. So in that case, I'm going to go back to the icon library, search nucleus. Let's pop in this nucleus here. And simply blow it up a bit more, like so. And that's going to be where you have a signaling pathway potentially that ends up with some transcriptional changes in the nucleus. And the nucleus, as you see here, has a brush icon in the icon library. So what that means is that it's actually, if we go over to the brush tab here, it's one of some of the more exciting icons we've added recently that are absolutely dynamically editable. So as you can see here in the tutorial, they basically, the features respond to how you change the shape, and that's really cool. So for instance, with this nucleus, I can go to brush options in the toolbar. Okay, I'm gonna close the tutorial here, and literally dynamically change, how small the subunits are in this kind of dynamic brush. And I can change, of course, the icon color as so. And just to showcase a couple more things here, going down to membrane brushes, this is something you can do. So drag out a membrane brush. Looks pretty similar to the top one, right? Except you can actually nicely shape exactly the kind of membrane shape you need. And our application would perfectly refit all the individual components so that it doesn't look janky or squished or anything like that. I'm gonna delete that. Okay, so next, just to finish up this quick figure before I, hand it over to Shiz to show more advanced features, I'm just going to enter a few maybe simplified proteins in the cytoplasm here of the bottom cell and connect them with some arrows. So if you go to insert shape here in the toolbar and scroll down to text shapes, here we have pre-made text shapes. So for instance, let's say I want to add some proteins. So click protein and then just kind of drag it out here. And there we go. I'm going to zoom in a bit. So I'm going to hold down Control and scroll wheel to zoom in. Here, pretty basic functionality. You can change how much these pills round by this little node here. And you can go to color presets and change from one of our curated color schemes. So let's go with this color. And I'm going to duplicate this by holding down Alt. Let's say protein here. There we go. Of course, you can edit text by double-clicking in as you need. And the bottom one, let's go with more of a blue. Okay, simple as that. And let's say now we add some lines. And we're really proud of how we've been able to build the lines tool. So I'm going to go to arrows, connect the signaling pathway. As you can see, very robust editability for the line tool. You can go through these tutorials on your own time when you jump into BioRender. For the time being, I'm going to go with this more faded dynamic curved arrow. So I'm going to click that and just drag it out from the receptor, the T-cell receptor at the surface to the protein A. And drag the protein A down a bit. And as you can see here, it has some nodes. So these blue nodes allow you to precisely change the trajectories of every line, like so. And then you can do a whole bunch of customizations. The tail arrowhead, the head arrowhead, the line type even. You can retroactively switch between different line types. But for the time being, maybe I'll just give it a very mild dash, make it a bit wide, a bit thicker, maybe not that thick. Let's say 3. And just make the arrowhead a little bit smaller, like so. That's perfect. I'm going to keep the line fade, as you can see here, just because it kind of looks nice. And I'm just going to duplicate that down like so, connect the arrowhead back to protein B. I'm going to remove this arrowhead by right-clicking and deleting node. There we go. And I'm going to duplicate this again and just point it into the nucleus somehow, like so. Perfect. And I think the last thing I'll show here before I hand over to Shiz is just a couple other very minor design things. For instance, to show these steps, a really great way is just to kind of label them with numbers. So if I go back to insert shape, text shapes, just pop in this little one here, click, there we go. Of course, fully customizable if you so choose. But I'm going to go with the black text, white background. A little bit smaller. 1, 2, and 3. Double click, edit the numbers. And you can annotate them by inserting some text. Drag out the text box here. Step 1, lorem ipsum A, and etc. Great. Okay, so if we do a bit of a preview here, here's where we got to. Hopefully I'm able to showcase some basic functionality in a slightly new light. Just getting everyone's brain thinking a bit about design, about how to, use these different customization tools to clearly and effectively communicate your own figures. Awesome. Okay, so I'm going to pause here and click share with to share this particular figure to Shiz. So I'm going to send a copy of this. And I think her demo email is shiz plus demo@biorender.com. Shiz, I don't know if you're on the other end of the line. Is this correct? Shiz
That's
plus
right.
demo? Okay,
Exactly. Thanks, Jerry.
great. So I'm gonna enter that, and please take it from there and send. Perfect. Okay, so without further ado, I'm gonna unshare my screen, and Shiz, if you wanna take it over.
Awesome. Will do. Okay.
Actually, I don't know how to unshare my screen. Whoops.
Let's see if I can just force
There we go.
Okay. Great.
Perfect.
Can you all see my screen here?
Yep.
Okay, fantastic. Thank you so much, Jerry. That covered a really, really good foundation for a lot of our features and a nice little history lesson. So thank you. So I've got my browser open. Jerry has sent me that figure that you just saw him compose. I'm going to actually refresh my browser just because it probably won't register until I refresh. And perfect. There we go. As you can see here, it's the last image that I guess shows up on my account here in my gallery view. And this little paper airplane means that someone has shared an image with me. And then just kind of like if you have Facebook or Twitter or something, the little red notification badge means I haven't opened that figure yet or modified it. So that's what that little red dot means. But it looks like Jerry's pseudonym, which is Ricky Ribosome, has sent me a figure. If I want to dig in and read that message, I can say view message, and there it is. There's that message he sent me. So I have some more context about what it's about. Sometimes it's about a publication figure or a presentation image, and it's got a little bit of notes like, this is version 5 of 10 or something like that. Anyways, I can actually now open this figure, and just so you know that any edits that I make on this copy will not be affected on Jerry's side. So it looks like he's gone and locked this, so I can kind of unlock this object. Maybe if I don't like how he's left some stuff overhanging here, I can clean that up. Same thing goes with this. I can, let's see, I can probably just crop it as well. That's the beauty of the brush feature. Anyways, yeah, I can make edits to this file, obviously fully editable, same way as if it was a template, essentially. Okay, so just so you all know that, I'm gonna go back to my gallery now. That little red button's gonna disappear, that little notification badge, but the paper airplane remains because, again, it's notifying me that it was originally sent by somebody else. Okay, so that's kind of how the share function works. I think it's quite powerful when you think about the capabilities of not having to attach it, in an email, like a PowerPoint slide, and then you kind of forget which version was the last one. This is a really elegant way to do that. Okay, so I'm looking at the concentrations of all of you who filled out the poll. Thank you for participating. So now I have a little bit of an idea of maybe your backgrounds, your fields of research or study. Maybe I'll go through our templates here as Jerry has kind of beautifully laid out in the introduction. And maybe I'll pick a template to start with. So I saw some cell and molecular biologists in the room, maybe some biochemists. If I type in the word cell, pretty broad term, of course, but you can see the variety of figures that show up with cell as a search option. Let's see here. I guess on the topic of maybe protocols, I'm sure a lot of you have to put together figures that show stepwise how you got to a certain conclusion or result. So, even if it's not a figure that you need exactly, sometimes what I like to do is just grab an image, take inspiration from it, and then maybe modify a couple of steps here and there to suit my needs. So here's one related to protein peptide mass spectrometry. I'm going to delete that title. Maybe I don't want the title in there. Kind of rejig a couple things. This is, sample data, so I'm just going to delete that. This might not be the machine I'm using. Maybe the protein mixture, I want something a little different. Maybe I don't deal with the brain, although I did see, I think, a couple of neuroscientists in the room. Maybe I'm working with the liver or the heart or the spleen or lymph node. Maybe it's, cardiovascular systems. Maybe it's embryology. Maybe it's even a mouse brain. Maybe it's a microbe. If I type in mouse, got a bunch of mice icons here. So I just want to kind of highlight how modular BioRender is. So say, for example, I have this white mouse. Let me move in here. And that's not the right phenotype. Maybe I need a dark mouse or a black mouse. So I've got that. Maybe I need a tumor. So again, very modular. By the way, if you didn't know that you could pick the color right from the library, all you do is click it and you can actually pick the color here. And I'm going to drag it out. Hopefully that matches. And there we go. Mouse tumor sticker kind of thing. And again, you can also change the color once it's in situ, but there we go. That's the mouse. Maybe we need some sort of needle. We have a lot of beautiful glassware as well. So we actually have a dedicated technical illustrator, you can call her, that actually creates all of these very beautiful technical illustrations. And then we've got biological and medical illustrators kind of making all of the more organic shapes and model organisms and such. Obviously the scale is a little bit off. It's probably a giant mouse if the needle is this small in comparison, but just for the purposes of our demo, I'm going to keep it that size. And sometimes you can skew reality to suit your image. You can take some artistic license. People will get it. They're not going to complain that your mouse and the mouse is huge compared to the needle. And maybe I don't like this straight arrow. Kind of like Jerry alluded to, it's nice to use dynamic arrows to kind of show a little bit of scale change, maybe motion. This is cool. Sample. Okay, I'm going to need maybe a dish, petri dish of some sort. And this is kind of cool. Our artists have actually put together a few grouped icons. Again, as Jerry showed, you could double click to edit this. And I kind of want to show that the cells match the color of the kind of liquid that I'm dealing with. Maybe the agar plate's also a slightly different color, like so. So that kind of matches, 1-to-1. Protein mixture. Maybe I can use one of our generic proteins if I kind of type in a protein. And this is weird. It's showing templates, and that means I'm kind of stuck in the template mode. So we're just going to toggle over here to icon mode. There we go. And those were where those generic proteins showed up. You can kind of just select these, I guess you can call them generic, but kind of blobby shapes. Sometimes you want just like an amorphous kind of a shape to identify if you don't want to, use the exact PDB ID. But that being said, if that is important for maybe the biochemist in the room or structural biologist, you can actually use our PDB plugin. And Jerry alluded to that already, where you can kind of create your own protein. We have a couple of sample proteins there ready to go. So if you click one, it'll automatically open to that. But if you have a favorite protein, of course, type in that alphanumeric, load it up. It's going to plug directly into the protein data bank, and you'll be able to color it any way you like. Maybe I'll use this kind of bimolecule shade. And what's really popular right now is this kind of toon shader. Looks like I'm using a real 3D rendering program. So save as new icon. Actually, Jerry helped to launch this new feature. So really cool. Great job, Jerry, on this. But again,
Thanks,
if you need if you need a little more high fidelity or structural kind of crystal structure related to that protein, you can use that. For now, I'm not going to use that. I'll put it off to the side there and use the generic proteins. Oh, one thing I'll note is that sometimes people will ask, how do I make my own proteins if I want something amorphous but not exactly the PDB? Because our proteins are all pretty modular, you can probably compose something with a bunch of our shapes, again, because we have exact color matching. So this yellow protein, you could turn to maybe a green, this one as well, and then maybe overlap them or something like that, and then group it. that's how you could maybe hack it to do, you know, kind of your own modified antibody there. But really, again, everything's meant to be modular, where we'll try as much as possible to give you color options that can match each other. Okay, so there's that kind of random protein mixture. Again, you can change the color later as you see fit. All right, moving along down here, let's find our machine that we need, mass spec machine. And if you've got a paid subscription, you can actually request a custom icon. I believe you need at least a minimum of an annual subscription if you're on the student plan. Also a lab license or an annual academic license, but then you can kind of go ahead and basically request anything under the sun or under the umbrella of life sciences if we don't have it already. So that's this little form here, again, as Jerry alluded to. So maybe this is the machine that I want, not the one that was previously shown. So I'm going to drag that out, resize it. And then finally, input my own data. I've actually pre-populated my uploads tab here with some data that I've exported from GraphPad Prism. I don't know if some of you use Prism to visualize your data. Oh, it's a little big of a file. You can see though that when I export it as a PNG from Prism, it kept that see-through kind of background with the figure. So you can see here that it's taking on that gray color. It's because it imported as a PNG. So that's really nice. If you have maybe even a logo of some sort that you wanted to import without that colored background, it's possible to do so. Here's this BioRender logo. Same deal. Doesn't have a colored background, so I can kind of overlay it on things and it'll take on that background color. And again, that format is called PNG. So there's my data kind of here, imported to finish off my figure. Again, protocol, these little numbers, we're using that number system that Jerry had shown earlier. So definitely take advantage of that because it'll lay down a perfect circle with that number in it. Really helps to draw the eye around the page. There we go. And say I'm happy with this figure. Let me click preview. Looks pretty good. And I want to now export it. You're probably going to use it for, you know, a PowerPoint slide deck, maybe a Google slide deck, or just you want to export it and, you know, share it with a friend or colleague or your PI or student via email. What I would do is click export. And again, same deal here. You can actually select PNG with a transparent background. I believe that's one of our premium features. JPEG or PDF. And we've got a couple of resolutions here. Generally speaking, I like 150 dpi for presentations. That just tends to be enough for my needs. And I'm going to go ahead and select JPEG for now. And what that's going to do is it's going to export and download to my desktop. Okay, and what I'm going to do is actually share my entire screen. Actually, no, I'm going to share my PowerPoint page so you can see that. Okay, and I'm going to do is basically what you can probably surmise I'm going to do is just basically download it right from my desktop. Go, here's the image, and nice and sharp, presentation formatted. There we go. And then sometimes we get asked, you know, what if I want to animate in BioRender? We actually don't have native animation built out yet. But what I do see some people doing is actually bringing in sections of an image or creating copies and then importing those so you can kind of advance your slide and then kind of animate those steps accordingly. It takes a little bit of you know, matching up and finicking, so don't recommend it necessarily unless it's really important for your storytelling. But this option is there for you if you'd like to use that, you know, transitional animation option. Okay, so I'm going to go ahead and share my Chrome browser again. There we go. We're back to the figure. All right. So that's how to create kind of a protocol from a template. Now, just to finish us off here, I wanted to go through perhaps maybe a cell-to-cell interaction figure. And so here's one that I've kind of prepared ahead of time. But again, really, really simple diagram. If I were to have made this from scratch, it would be super easy. Type in cancer cell. There we go. And again, this is just to show you that everything's very modular. Send to the back. And one, I guess, tip that I'll leave you with that I think we see as one of the biggest mistakes seen in figure making is often the level of contrast in a figure. So say this was perhaps a different color tone, like a green. If there happens to be any colorblind folks in the room today, you'll probably notice that that disappeared pretty intensely. So what you can do is instead of trying to memorize what is a colorblind friendly color versus not, what you can actually do is use our handy preview and grayscale mode in BioRender here. So it's under the view, drop down preview and grayscale. And what this does is it basically eliminates color as a variable. And so if we live in a color or world of just grayscale or black and white, you'll know right away that there are certainly areas of figures that are breaking some contrast rules. So here we go. This double-stranded DNA now looks like RNA or single strand. Zooming in here a bit, because that second strand is just so close in contrast to the nucleus color. And this is a challenge because, nuclei stain dark. So a lot of things that you overlay on nuclei are going to be naturally a little bit hard to register. So what I would do is just punch it up by making those foreground elements really dark. You can even roll over our color options here and it'll live update the contrast. There we go. And then same goes for this apoptosis kind of label. A couple things you could do here, and you can probably guess it. I can go extreme by making that label white, or maybe a little bit more of a favorable option, I'm going to undo that, is to make that cell color a little bit less opaque, maybe about there. And again, we're in kind of black and white mode, so you're kind of safe to play around with these toggles. This might even serve well to be a little bit less strong as well. Now, if I go back into full color mode, there we go. We haven't lost the cells. They're still there and they're still registered, but those foreground elements are much clearer now because we've knocked back the opacity. Another trick I like to use is when you have kind of like a spray of cytokines or interleukins, whatever it is, maybe it's antibodies flying around. It's nice to show this directionality where it goes from concentrated to unconcentrated like this. This isn't a grouped icon, so maybe I'll look for one that is. Just put in dots. Here we go. This is a nice one. This is telling me that I can probably go in and edit some of those dots. But that's a trick, if you want to show directionality of the source of all of these sprays, you can certainly use that directionality. And then also going back to our trusty arrows, you can tell here that medical illustrators love arrows, is to use that faded curved line instead of those harsh lines like this. Sometimes you'll notice some figures showing like just a thick line like that, showing directional movement. Try to avoid that and use a little bit more of a softer curve to your arrows. And I obviously would thicken that line so you can see it. You can change the arrow head size. So I'm gonna knock it down a little bit. And then that black line's looking really harsh. My eye's going right to that part of the picture. So I'd probably just even color pick. And if you look at the top left corner, you see that color block changing as I scroll over things. It's going to color pick whatever it is on my canvas that I need to color pick from. So maybe it's this dot. There we go. It's looking pretty good. And then of course, if you want to get really fancy, you can always knock back the opacity of the arrow itself just to soften it up a bit. Okay. So a couple of kind of 5-in-1 design tips there on how to tell stories a little bit better. And of course, make sure that you have enough contrast in your figures, because this is a universal issue that we see across any figure making endeavor, is the lack of contrast a lot of the times. Okay. I'll come back to my gallery here. You'll also notice that that picture kind of auto-saved as well as the ones previous. If I want to recover a previous version of a figure because, I don't know, my browser crashed, or perhaps I just don't like what, design decisions I've made, I can actually go back in time and revisit that figure from long ago. So let's see, this is May 2020, and it looked quite different then. But if I make a copy of this version, now I'm going to have 2 in my library. So this happens a lot. Our internet connection might, bug out or something, and you might want to recover a previous version. That's the way to do it if you ever get stuck in that situation. Okay. Great. So we did promise a little bit of time at the end here for some Q&A. So maybe I'll just toggle back here to Jerry to see if there are any outstanding questions, and then perhaps we can go through any kind of plan or pricing questions we have in the rest of the slide deck.
Thanks, Shiz. I think a lot of the questions from today were about a bit more peripheral things, like how much, what is the pricing plans, what are the pricing plans like, and what are the publication aspects to figures made in BioRender. Yeah.
Sure. So maybe it would help to now switch gears to show that slide then. Okay, I'll go ahead and stop sharing.
Let me things over and share. Okay, cool. Oh, whoops. A few other slides to, I guess, show some cool figures that some previous BioRender users have been able to make. But in terms of pricing, that's a great question. I think one of our attendees asked a bit earlier in this presentation. We do provide a range of premium options. Of course, everyone here, if you haven't signed up for a BioRender account yet, it is free forever. So you can sign up for an account, check things out. All the icons are available for all users, including free users, as well as I would say 95 to 98% of all the editing functionalities as well. That being said, there are boons to signing up for monthly or annual subscriptions. For instance, one of the major ones in academia is publication rights. So you have educational rights to use any of your BioRender created figures as a free user. So posters, theses, dissertations, lab presentations, departmental talks, those are fine. But if you would like to publish a figure you've made in BioRender in an academic journal, then we have these rights provided in, for instance, the academic individual plan, which is $35 a month USD on an annual plan. Alternatively, if you or someone you work with is a graduate student, so all the way up to PhD student, we do have a heavily discounted graduate student promotional plan, which, if I remember correctly, is $15 per month on a monthly plan. And of course, if you would like to save a bit and you would like your whole lab to be on an academic plan, then as we show here, there's a potential for 5 multi-seat or 5-seat kind of multiplayer plan at $100 a month. And lastly, we are trying to, as a company, work with various academic and industry institutions to, do these subsidized institutional plans. And if that's something you're curious about, or you'd like to ask about, or that you know someone at your department or your institution as a whole you'd like us to talk to about, just contact us in our BioRender.com/pricing page. Yeah, so hopefully that covers both of those questions. If anyone has any additional questions about anything at all, including these more administrative or pricing related questions, we have an amazing support team who you can access in the BioRender application, in the kind of like chat bubble that you can click, or at support@biorender.com via email. Okay, I think that wraps up our webinar presentation today.
Yeah, thanks
Shiz,
so
do you want
much,
to add a few
Jerry.
more words?
No, I think you wrapped it up very nicely. I guess a granular question we got in the chat was, how do we get borders around individual figures in a template? I'm not maybe quite sure what that is referring to. Perhaps it's related to how do you create a border around a figure as far as the canvas. You can certainly maybe create a box and put it in the background, and then that way you'll create that border. You can create boxes with no fill as well in the shapes panel, so that might help with what you're looking for. Hopefully that answers your question. Otherwise, please feel free to write in to support@BioRender, and we can try to get that answered for you. And yeah, if anyone else has any, okay, great. If anyone else has any design related questions and you think of them later, please feel free to reach out. As Jerry mentioned, there's a little chat function in the bottom right of the app that you can do so.
Great.
Yeah, that's everything from me. I guess we can wrap up here.
Yep, sounds good. Okay, thanks everyone. Hope
Thanks,
you had
everyone.
a good time. Have a great day.
Bye-bye.
Bye everyone.